Hi, everyone. Welcome to our final University Scholar Series presentation. We're here today to listen to Dr. Brigitta Mcdonald from Moss Landing Marine Labs share the impacts of disturbance on marine mammals, physiological and behavioral responses to stressors. I'm Magdalena Barrera, Vice Provost for Faculty Success, and on behalf of Provost El Casino and the team in the Provost office, I'm really happy to be able to welcome you to today's event. And before we begin, I'd like to start with the land acknowledgement. While we gather here at San Jose State University, we're gathered on the ethno-historic tribal territory of the Tamian Ohlone, who were the direct ancestors of the lineages enrolled in the Muwekma Ohlone tribe and who were missionized into Mission Santa Clara, San Jose, and Dolores. The lands on which San Jose State University is established was and continues to be of significance to the Muwekma Ohlone tribe. We also recognize that the ancestors of the Muwekma Ohlone constructed and maintained 3 Bay Area missions. Our campus extends to the surrounding areas that held a tupintac, a traditional roundhouse, which were once located at the historic Lope and Yigos land grant Rancho Polsomí y Positas de las Animas, Little Wells of Souls, and also Marcelo, Pio, and Cristobal's land grant Rancho Listac, which were places of celebration and religious ceremonies, as well as nearby ancestral heritage shell mounds that served as the tribe's traditional cemetery sites and territorial monuments. San Jose State University also desires to honor the military service of the Muwekma who have honorably served overseas during World Wars I and II, Korea, Vietnam, Desert Storm, Iraq, and who are still serving in the US Armed Forces today. So before we get to Dr. Mcdonald's talk, we want to do a little bit of housekeeping. And that is first, I want to remind you that live captioning is enabled. So please take advantage of that feature. This webinar will be recorded and shared later with folks who couldn't make it here live. And there's also opportunities for those who are able to attend here right at this moment to ask a question. We ask that you do that through the Q&A function and to please make sure that all attendees are selected when you submit your questions. And we'd also like to thank the number of campus partners that help us make the series happen. So that's the Office of the Provost, King Library, the Department of Moss Landing Marine Labs, Spartan Bookstore, and the Division of Research and Innovation. Thank you all for your partnership for this series. And now again, it's my honor to be able to introduce Dr. Brigitta Mcdonald. Dr. Mcdonald is an associate professor at Moss Landing Marine Labs, where she's been teaching courses on ecology and physiology of marine vertebrates and scientific skills since 2015. Dr. Mcdonald obtained her doctorate at the University of California, Santa Cruz, and conducted postdoctoral research at Scripps Institution of Oceanography and Aarhus University in Denmark. She also holds a master's in biology from Sonoma State, where she was recognized as a top 25 scholar, and a bachelor's in biology from UC Santa Cruz. Dr. Mcdonald is a physiological and behavioral ecologist. Using state-of-the-art biologgers, her work investigates the adaptations that allow animals to survive in extreme environments. Now, understanding the mechanisms that allow an organism to interact and survive in its environment is crucial for predicting how they will respond to climate change. So in this regard, Dr. Mcdonald's work is essential for addressing the significant climate challenges that we face in our future. Through her research, Dr. Mcdonald has worked with a broad range of species, everything from emperor penguins, sea lions, elephant seals, deep diving toothed whales, dolphins, porpoise. It's like a greatest hits of all the really amazing nature shows that we get to see on TV. In a diversity of habitats from the Antarctic to the Galapagos. And so with that, let's get started. And Dr. Mcdonald, welcome. And I'm very pleased to turn it over to you. Thanks.
Thank you very much for that nice intro. All right. Well, I'm excited today. Oops, my slides are jumping ahead of me already. So I'm excited to be here today to talk about some of the research that my lab has been doing, where we're studying the impacts of disturbance on marine mammals. But before I get into some of the different research projects, I thought I would just do a quick introduction to the type of research that my lab does, which you've already just been given a nice introduction to. So I consider myself a physiological ecologist, which means I study how animals are physiologically adapted to survive and reproduce in their environment. And the reason I was really drawn to this field is because basically every class I took as a student in biology, so ecology, evolution, physiology, I loved them all. So I didn't really want to have to decide what to do. So the field of physiological ecology allows me to kind of continue working in all of those different areas. Most of my research is focused on marine mammals, and they are a model system for a physiological ecologist because they have a huge variation in size. So they range from small Antarctic fur seal pups of about 5 kilos at birth, 4 or 5 kilos, up to the largest mammal, the blue whale. They also are found in a bunch of different types of environments, ranging from the cold polar regions to more warm tropical waters, such as up in Ecuador or in the Galapagos. They also have different evolutionary histories. So the different groups of marine mammals have kind of made the transition back to the marine environment at different times. And so they're great for comparative studies. And then as a physiologist, they are ideal because they face physiological challenges on a daily basis. So every time that they dive down to get fish, they have to deal with the lack of oxygen or hypoxic challenges. Many of these species migrate, they might undergo long periods of fasting while reproducing, so they face energetic challenges. Those are the two kind of primary areas that my lab focuses their research. So one big focus is diving physiology. So how do these breath hold divers, these marine mammals, how do they dive down to great depths? How do they hold their breath for so long? And then the second area is energetics. So how much food do these animals need to meet their energy requirements, and how do they allocate it towards different things such as growth, reproduction, and other behaviors such as continuing to forage. I ask these physiological questions in an ecological context. So I want to understand how their physiology kind of constrains their behavior in ecology. For example, how does their diving physiology constrain their ability to forage and find food? By doing this, you can get a better understanding of what the ecological niche of an organism is. So what's their role in the environment? And more recently, I've been trying to ask these questions with a little bit of a kind of conservation view. So kind of how is their physiology going to influence how they respond to some of the challenges that they face? So some of the biggest challenges that they face, where it's really important to understand kind of their physiology, is climate change. So if there are changes in prey quantity or prey location, that can present them with energetic challenges. How are they going to meet their energetic needs? There may also be some thermal challenges. There are also increases in disturbance in the marine environment. So as our population has been increasing, we're more on the coast, there's more activity, human activity out in the ocean. How are they going to be able to deal with these disturbances? So there could be acute responses, which might be an immediate change in behavior that may reduce their foraging success, or it may increase how much energy they're using. There can be stress responses, so they can have elevated cortisol. Again, stress responses, they exist for a reason, but having them for extended periods of time can be detrimental to the animal's health. And there may be, direct injury or illness. Some of the responses may be more chronic. So if there are continued lost energy or kind of energetic challenges, they spend more energy or missing out on feeding opportunities, this may result in animals being in poor condition and ultimately leading to decreased reproduction. So you might be asking yourself, why do you care about marine mammals, or why should you care about marine mammals? They're really important for helping to maintain a healthy ecosystem. So they're top predators. So they help keep prey populations and basically everything lower in the food chain in check. Additionally, some of the smaller species are important prey items for other species. And then more recently, there has been a talk about how they're very important for nutrient recycling. So a lot of the nutrients from kind of the upper ocean will sink down into the lower ocean. The whales and other deep diving animals that feed lower, kind of below where most surface animals can reach, they can bring those nutrients up to the surface so they can eat at depth. And then when they poop in surface waters, basically they are bringing those nutrients up. The phytoplankton need that to grow. Then this can actually be transported by seabirds and fish into the terrestrial environment. It's actually been predicted that whaling, kind of the decrease in marine mammals over, when they were hunted more extensively, reduced how much nutrients were being brought up to the surface and accessible to other organisms by 77%. So they are very important in this nutrient recycling, and as their populations are, or many of them rebounding, this is very good for our ecosystems. Also, marine mammals are very important in some locations for economics. So whales are a big ecotourism attraction, and this is important for many different locations. And then in general, marine mammals are kind of the characteristic marine megafauna or charismatic marine megafauna. So they're cute, people are interested in them, and so they can be a very important or a very powerful tool to engage people in environmental issues. So in today's talk, I'm primarily going to focus on disturbance and how these animals respond to some different types of disturbances. So when an animal is exposed to a stressor, this can be a disturbance such as an acoustic disturbance, human interaction. It could be as a result of climate change. Maybe there's less prey, there's more pollution. They may exhibit a physiological change, so a stress response, increased cortisol, or a heart rate response, changes in metabolism. And they may also experience a behavioral change, and they're likely related. These types of changes over extended periods of time can have a chronic effect on the health of the animal, so their condition. And if they're in lower condition, this may make them more susceptible to disease. So it's really important that we kind of understand how they respond, what the physiological response to stressors are, what the behavioral responses are, and linking this to animal health. Some of these changes can have more direct acute responses that affect the vital rates directly. So basically the health and also the acute responses may impact the survival, the fertility, and the growth of individuals. And ultimately, this will impact the population. So today I'm going to primarily talk about more of these acute responses and then start to talk about a study or introduce a study that we're just starting that will look more at the chronic, but we're really just trying to kind of fill in some of these squares here so we can better understand how disturbance is going to impact populations. So the two kind of common types of disturbances that I'm going to talk about today is the first is tourism or human interaction. So even though ecotourism can be great, we need to do it safely. And I'm going to give a case study of sea otters. So look, again, I'm using primarily local species in today's talk. And then the other type of disturbance I'm going to talk about is noise in the ocean. And I'm going to be using elephant seals, a study we're doing with elephant seals to examine that impact. So I'm going to start off with kind of a local example, a research project that was led by a previous graduate student, Heather Barrett, in collaboration with Sea Otter Savvy, where we looked at what is the energetic cost of human disturbance on the southern sea otter. So the issue is basically sea otters are very cute. They are a coastal species, so they're easy to observe from land. They're easy to get to in kayaks and small boats, and they're found in populated areas. So there's a lot of opportunities for these animals to be disturbed. So we know that these animals are experiencing high levels of disturbance. So this makes them kind of a model species to look at what are some of the potential costs of disturbance. Otters are also ideal because they spend a lot of their time at the surface. And like I mentioned before, they're close to the shore. So you can observe them from the side of the road or from the coastline with spotting scopes. So we can see how do they respond to these disturbances. And because their metabolic rates have been measured in captivity, we're able to link this change maybe from sleeping to rapidly swimming away. We can link these changes to measured metabolic rates to estimate what is the cost of this disturbance, what's the energetic cost. And otters are thought to be often operating kind of at their energetic edge. They are one of the smallest marine mammals that live in cold water. They have very high metabolic rates. And so how is this potential additional cost going to kind of impact these animals? So the goal of this project was to first determine how often are these otters disturbed, and kind of how disturbed are they? How much does their behavior change? The second goal was to look at, well, what are some of the factors that may influence how the sea otters respond? And the third is if there is this response, what is the energetic cost? So this study was conducted at 3 locations in the Central California coast. So Morro Bay, Monterey, and Moss Landing. And a group of volunteers, along with a graduate student, would perform these 2-hour behavioral observations where they would record what the otters were doing. So it could be something like resting, alert, grooming, swimming. They would record any potential stimuli within the area, kind of in the view. This could be kayaks, paddleboards, different size boats. They recorded how close the boat got to the otters. They recorded some of the environmental variables that might influence response, such as tide, wind, and how much kelp was available in the area. And also just some general information about who was there, like what otters were there. So how big was the group, and were there any pups in that group? In total, they got over 1,300 hours of observation. And all of this data then was kind of fed into these hidden Markov models in order to see kind of how often are they disturbed, and what is the cost of this. So this is one of the kind of primary outputs that she got, and this is showing a curve. Basically, how does the distance of a stimuli influence their likelihood of being disturbed, so the potential for disturbance. And so this is a curve for a small vessel, things like kayaks and paddle boards. And what this is basically showing you is that when the stimuli, the small vessel, is further away, there's less than a 10% chance that the sea otter is going to alter its behavior, that it's going to be disturbed. But when that kayak gets closer, so maybe within 10 meters or about 30 feet of the otter, there's a 50% chance, or greater than 50% chance, that the otter is going to be disturbed. So you can use this to kind of estimate how close should people be allowed to get to the otters. One of the interesting outcomes, so this is the average, is that this varies by location. So in the Monterey Bay area, otters seem to be a little bit more sensitive, versus in Morro Bay, they might be a little bit more habituated to the boat traffic in the harbor. They were less sensitive. So on average, so kind of typical guidelines for viewing marine mammals encourages people to stay greater than 50 meters away to avoid disturbing them. And if you look at our average model, that works pretty well. There's less than a 10% chance that you are going to disturb an otter if you're more than 50 meters away in your kayak. But in Morro Bay, because they seem to be a little bit more habituated, you could potentially get a little bit closer. But in Monterey Bay, if you go to that same distance of about 25 meters, you're very likely, there's a 30% chance that the otters you're observing are going to be disturbed. So it's one of those things to keep in mind. This average curve is pretty good for setting some general guidelines, but there are going to be some differences between locations. So it's probably good to err on the side of caution, and that 50 meters is probably a pretty good rule to follow. And so she was able to create this curve basically that says, that she can share with stakeholders, policymakers, that shows, okay, you can maybe set this different distance. If you say 30 meters, there's less than a 20% chance you'll disturb the otter. So you can decide, well, once we know how costly this is, kind of how should we set these regulations? So this was information that she has been sharing with, or the Sea Otter Savvy has been sharing with different groups, ecotourism groups in the region. So I'm going to skip the second objective. There wasn't a lot of evidence that the variables we measured, the kelp, and the presence of pups influenced the level of disturbance. So we're going to just skip ahead to that modeling that energetic cost of disturbance. And so what I'm showing you here is all of the observations we're taking during daylight hours. So we're just showing, and that's usually when most of the disturbance would happen, what is happening between 6 a.m. and 6 p.m. And based on all of the observations that they took, you can see they can model, well, how likely is an otter going to be active? So otters spend a lot of time resting. So they're either usually out feeding, grooming, or resting. That's kind of their most common behaviors. And in Monterey, so this is using Monterey as a location, following a single otter where there's, the ratio of 50% pups. So 50% of the females would have pups. 50%, there's 50% kelp cover. And you can see that there's about a 30% probability that an otter will be active throughout most of the day. Well, what happens if you add disturbances? So based on all of those observations, they know in Monterey, a typical day will have, sorry, went the wrong way. We'll have about 6 different disturbances where kayaks come within 20 groups of, or 20 meters of a sea otter. So this is the kind of the typical level of disturbance to a sea otter in Monterey. And you can figure out what the cost of this disturbance is by kind of converting. You can see that there's this difference here, when the animal is disturbed versus not disturbed. And you can take that activity level difference and convert that to energy expenditure or kilojoules. So how many calories, basically additional calories, do they need to eat in order to make up for that disturbance? So when she did this for an adult male, kind of average size, she found that in a typical day in Monterey with 6 disturbances where a small vessel approached to within 20 meters, a male would either have to eat about a little less than a half a crab more, or almost 11 Pacific little neck clams, or 19 tegula, little marine snails, in order to meet this increased energetic requirement. So it doesn't seem like it's necessarily that much on a typical day. These are typical, some of the common prey items that otters in the region eat. And otters do tend to specialize. So an otter will often specialize on crab or clams or tegula. So this is just kind of giving you an idea of how they might have to alter their behavior. And so even though this may not seem like, well, how hard is it to get 12 more clams? Some days there are going to be more disturbances. Maybe sometimes there's less food availability. And then another issue, there are some hidden costs that we're not measuring. So if an otter now needs to eat 10 more clams, he has to spend more time foraging to get those clams. So that's less time resting, and then spend the energy diving down to depth to get the clams. So there's a lot of unseen costs associated with this. But basically, what she concluded is that the degree of response depends on the location. So Morro Bay animals seem to be a little bit more habituated and less responsive, and also depends on how close the stimuli gets. The external factors that we measured didn't seem to influence the degree of response. And we did find evidence that the typical disturbance levels resulted in just a small energetic cost, but it's really important to remember at this point, it's not the full cost. So research is continuing to go on to try to get a better idea of what the true costs are. And also there may be some unobservable changes. I'm sure some of you guys have felt stress before where your heart rate increases, but you may not alter your behavior. And that's something we aren't able to measure yet. Adult females are thought to really be operating close to their edge, kind of energetic edge. There's something called in-lactation syndrome where some otters seem to not be able to meet their energetic requirements. So they're also trying to get a better understanding of how reproductive females respond to disturbance, and if that could be kind of impacting, like pushing them over their energetic edge or limits. So the second issue problem that I want to talk about, or some of the, another issue, another type of disturbance that these animals face is ocean noise. So in some regions in the world, over the last 6 decades, ocean noise has been doubling every decade. This increased noise in the marine environment can interfere with the foraging capabilities. So many of these animals use sound to find food. It can also influence or negatively impact the ability to communicate with conspecifics. Also, increased noise in the ocean may increase the stress level of animals, and it may alter their behavior. So they may end up not foraging in prime habitat if there's a lot of noise there that's kind of deterring them, or they might have kind of one of the things we're going to look at, like an acute response to a disturbance that may cause them to behave in an atypical way, maybe swimming to the surface a little bit quickly, that can put them at risk for things like decompression sickness. So if there's any divers out there, many of you guys have probably heard of decompression sickness. If you dive and you're breathing air, you absorb a lot of nitrogen. And then if you surface too quickly, this nitrogen can basically come out of solution, be bubbles. Kind of think about like opening a can of Coke, the carbonation that comes bubbling up. And this can cause some health issues and potentially even lead to death. For a long time, we didn't think that marine mammals would suffer from this because they were just diving on one lungful of air. So they're not bringing as much nitrogen with them on a dive. But we now know when they perform repetitive dives, they can build up the amount of nitrogen in their tissues. And so then if they alter their typical behavior, they may be at risk to decompression sickness. And this was suggested when there were some mass strandings associated with naval sonar. When they did necropsies on these animals, they did find some bubbles in the vessels that suggested that they might have suffered from decompression sickness. So this is actually a photo from a turtle that had decompression sickness, but it was one of the nicer photos where you could see some of the bubbles where there should be blood. So this does suggest, even though this may not be typical for them to get decompression sickness, if they alter their behavior, they may be susceptible. And some of the factors that determine how at risk an animal is to decompression sickness are things like blood flow and distribution during dives, how much air they take with them on a dive, and when their lungs collapse at depth. And these are many of the factors that my lab kind of investigates to try to understand why some animals seem to be more at risk. So before I start talking to you about the impacts of an acoustic stressor on elephant seals, I first want to do a crash course in diving physiology. So this is one of my favorite things to talk about. I can talk about it for hours. You can just ask my students. I'm going to try to do it in just a few slides. So the field of diving physiology basically is studying kind of how do these breath hold divers deal with lack of oxygen? So how do they manage their oxygen on a dive? And then what do they do when oxygen levels get low? The other major area or research focus is how they deal with pressure. So many of these animals dive to great depths, 1,000, 2,000 meters. How do they avoid tissue damage? And how do they deal with some of how do they avoid some of the illnesses or problems associated with increased solubility of gases at depth, for example, decompression sickness. So marine mammals are breath hold divers, which means their food is at depth, but just like all mammals, they have to have air. So they return to the surface regularly. They should behave in a way that's going to maximize the amount of time that they can spend underwater foraging. And they can do this by diving aerobically. So about 40 years ago, Koyman and colleagues coined the term the aerobic dive limit. And it's defined as the dive duration in which you see an increase in blood lactate at the end of the dive, which means the animal has started to use anaerobic metabolism, so metabolism that doesn't require oxygen. This is data from that first study. Each one of these dots represents a blood sample taken from a Weddell seal at the end of a dive. So you can see that in dives that are less than 20 minutes in duration, lactate levels were really no different than resting, so they're diving aerobically. But once the animal started to perform dives that were greater than about 20, 21 minutes, you start to see this increase in lactate, with the longest dives having the highest levels of lactate. So this inflection point is what was defined as the aerobic dive limit. So as long as the animals dive for less than 20 minutes, they didn't produce excess lactate, but when they did, the longer the dive, the more lactate they produced. They then had to spend longer at the surface to clear that lactate. The reason this is important is because these animals want to maximize the amount of time they can spend underwater foraging. And they can do this by diving aerobically. So a Weddell seal with an aerobic dive limit of 20 minutes can make repetitive 20-minute dives and just spend a minute or two at the surface, reloading oxygen, offloading carbon dioxide. But if that animal, for some reason, performs a long anaerobic dive, for example, here, a 60-minute anaerobic dive, it will then spend, potentially more than 100 minutes at the surface recovering from that dive and clearing that lactate. So what this means, by diving aerobically, these animals can spend more than 90% of their time submerged. But if they start to perform long anaerobic dives, that can decrease to less than 40%. So, performing these these aerobic dives can really maximize their foraging efficiency, their time underwater. So marine mammals have a suite of adaptations that allow them to maximize the amount of time that they can spend underwater diving aerobically. One is they have a lot more oxygen stored in their blood and their muscle. And then the second is that they have mechanisms that reduce the rate at which they use this oxygen. So they have a diving bradycardia, or a decrease in heart rate. So they can decrease their cardiac output by up to 90%. You can imagine if your heart rate or your cardiac output dropped by 90%, you'd have very low blood pressure and pass out. So associated with this, these animals have peripheral vasoconstriction. So they can basically cut off blood flow from their muscle and really prioritize providing blood, oxygenated blood, to the brain and the heart. And this is just an example of some work with a harbor seal, where you can see the heart rate, went from about 100 and 150 at the surface down to about 20 beats when it was breath holding. And you can see this decrease in muscle blood flow. So now that we had this crash course in diving physiology, I can talk to you a little bit about how does this change when an animal is disturbed. So this is actually a project that we're wrapping up this year. I'm in the middle of the field season. I was working with a seal this morning, and after this talk, I will actually go right back out to working with the seals. We are studying how we're using at-sea experimental disturbances to characterize the physiological plasticity in diving seals. And this is a collaborative project with many different universities involved, and it's been a lot of fun. We're trying to come up with new plans how we're going to continue this research. Elephant seals are an excellent model to see how will disturbance alter their ability to manage oxygen, or maybe put them more at risk to decompression sickness. They're exceptional divers. They routinely dive to depths that are greater than 400 or 600 meters in depth. They perform long duration dives. So you can see this bout of dives. These are about 1 hour dives, and the animal's just spending 2 or 3 minutes at the surface in between. We know from previous studies that they have a behavioral response to noise. And probably what is most important, even though elephant seals aren't at risk, there aren't these mass stranding events, because they are exceptional divers and they have this behavioral response, we can instrument them with data loggers that measure things like heart rate and oxygen levels, and we can get them back. That's something that's not really possible with some of the deep diving cetaceans. We can't measure blood oxygen in those animals yet. So one of the goals of this project is to get a comprehensive picture of the dive phenotype. So basically link their behavior to their physiology. So we're using tags that measure their diving depth, their diving duration, and their activity. So we can see every time they stroke, their orientation, and we're linking this to physiological variables such as heart rate, muscle perfusion, and blood oxygen levels to really understand how they manage their oxygen on these long dives. And then we are adding an acoustic stressor. In this case, we're using a killer whale sound that alters their behavior. And we can characterize how does their heart rate, muscle perfusion, and use of oxygen change when they are stressed. We can look at short-term responses in behavior and oxygen management, and also more intermediate responses, such as measuring stress hormones. So to do this, and this is kind of what we're doing right now, we go out to Año Nuevo, which is a little bit north of Santa Cruz, and find some nice, healthy 1 or 2 year old elephant seals. We sedate them and bring them back to the lab where we can attach our data loggers. And then after they recover, we drive them down to Monterey and they get this nice ocean sunset release. And they swim back to Año Nuevo. And the reason this is such a great study system is because they cross the deep canyon. So we can get some great deep dives, and we can get our instruments back in about 2 to 3 days. This is an example of some of the data that we can get. So here, this is the depth profile. So this is a dive down to about 450 meters. In addition to measuring the depth, we are measuring how often the animal is stroking. So this is one axis of acceleration. So here you can see when it's closer together, they're stroking at a faster rate. Here we have heart rate. So at the surface, heart rate is about 120 beats per minute. It rapidly drops down to 50 at the start of the dive and continues to decline during descent. And it's usually somewhere around 15 or 20 during the bottom part of the dive before the animal starts its ascent and the heart rate starts to increase again. Last, we measured the blood oxygen in the venous blood oxygen in the animal. And so you can see that this is decreasing throughout the dive. When the animal starts the ascent and heart rate increases, there's often this little bit, which suggests there's potentially a little bit more kind of blood flow. And then we start to see it decline again. And in this animal, in this dive, we exposed it to that acoustic stressor. And rather than continuing on its ascent, we see this reversal, this dive reversal. The animal goes back down to depth. And what is most impressive is we see that heart rate drops from about 40 beats per minute down to 3 beats per minute. So for us, when we get scared, our heart rate often increases. Well, you can imagine for an animal that is carefully managing its oxygen, that's not a good response. And that's not what they do. They have a pretty extreme decrease and it stays low throughout a lot of the dive. Like it's normally getting much higher here. So it alters their heart rate response, so their oxygen management when they're diving. And you can also see that here. So when the heart rate plummets, they're isolating the muscle and you don't actually see any more decrease in the blood oxygen. They're able to kind of conserve some of that oxygen. And then you see the decrease once the animal gets closer to the surface. And to help visualize this, we worked with a graduate student at UC Santa Cruz to create this animation for us. And I just kind of cut some different segments. But before we get into it, I just want to highlight some things that you can watch in this 2-minute animation. So this tells you kind of where we are into the dive. So this is before the dive starts. You can always look up here to see at what depth the animal is. This tells you their stroke rate. And then here we'll have a running chart. So this shows the heart rate in pink. So at the surface, it's about 120. And then you'll see this white bar graph here that is showing the stroke rate. So when I first start, I'm going to show that the first part of this video will show the first 20, or will show until the animal reaches a depth of about 25 meters. And you can kind of see here the heart rate. And then we've included sounds. So the beats that you hear is basically each heartbeat. And I'll kind of walk you through some of this as we watch this. So these animals are very efficient swimmers. So the animal is stroking initially, but we've actually seen the last stroke for the entire descent. So he's now just sinking to the bottom. You can see the stroke rate is down to 0. The heart rate has gotten down to 40 beats per minute. So now we're 7 minutes into the dive. The animal is about 340 meters. Heart rate is still kind of jumping between 20 and 40. We've had no strokes since that last stroke at about 15 meters. So now we're jumping to the animals. We're 15 minutes into the dive. The animal is ascending. We're 270 meters, and then we're going to scare it. So you see the animal starts to stroke, the highest stroke rates that we've seen. And I'm going to actually be quiet so you can hear the heart rate. So now the animal is starting its ascent again. The heart rate is still much lower than it was at the same depth. We're 21 minutes into the dive now, so you can just see the animals stroking hard, heart rate's still pretty low. And now we're at the end of the dive, the heart rate's starting to increase. As the animal approaches the surface, it's positively buoyant, and so it just floats up to the surface. Yeah, so that is just kind of gives you an idea of the type of data that we're getting from these tags and how we're using it to understand how disturbance is impacting their ability to manage oxygen. So from this study, we're learning a lot more about what these animals are capable of and how they're able to modify their behavior when they're disturbed. It's also helping us to understand what are the mechanisms underlying their dive response. So how does exercise or how hard they're working influence, and how does things like fear or volition or planning influence their heart rate? One great thing about elephant seals is that we can measure a lot of different things at the same time, so we can improve our understanding of how behavior is influencing heart rate and blood flow. This is something that we won't be able to get from some of the more at-risk species, such as the deep diving cetaceans, where we're hoping to just get behavior and heart rate. And this is going to allow us to better predict how these animals respond to stressors and also apply this to more difficult to at-risk species. So I have been working with some colleagues in Europe to develop a tool where we can at least measure heart rate in response to an acoustic disturbance. So we're taking a DTag, which measures, that's previously developed tag that can record sound, acceleration, behavior, and we've been working to add a new sensor so it can also measure heart rate. We've had some delays due to COVID, hard to get out in the field in Spain, but we have tried out the tag on wild pilot whales. We've had 5 attempts, 3 of them are successful. So although people are quite good at attaching just the DTag, having this external electrode seems to increase more drag and the tags don't stay on as long. But we have gotten a couple hours of data that is starting to inform us on how these animals regulate their heart rate, and we're continuing to develop this tag, so hopefully we can get longer records and see how they respond to different sounds in the ocean. Overall, these tags performed well. When the animals were resting and swimming slow, we could pick up the signal in the acceleration. So you can see each one of these heartbeats here. We could also hear it with the speaker, and we recorded it with an EKG. So now we just need to get it to work when the animals are swimming a little bit faster and to stay on longer. So most of what I've been talking about so far has been focused on acute responses. I'm just going to end by talking about a study that we're just ramping up right now that will look at some more chronic responses. So how do stressors potentially impact condition and ultimately fertility and reproductive rate? So this is a study where we are assessing the impacts of multiple stressors on marine mammals using elephant seals as a model species. Like I said, this also had some COVID delays, but we were ramping it up this year. So we have 23 animals out now collecting data. And so what the stressors we are using is we're injecting these animals with ACTH, which is the hormone that stimulates the cortisol release. So we are stimulating some of the animals before they go to sea, so they're leaving with kind of an elevated stress level. We are also instrumenting them with the acoustic tags, like we used on those juvenile seals, and we'll expose them to a killer whale sound about 3 times a day. And then we're also selecting animals that have varying mercury loads. So we're trying to see how these multiple kind of potential stressors are influences their foraging success and their health status. So this last breeding season, we instrumented 6 control animals, so no manipulations. 6 animals were injected with just ACTH to increase their stress response. 6 had the acoustic tags, and another 6 had the ACTH tags. In total, we're hoping to do 72 animals over the next few years. We can look at their fine scale foraging behavior and then look at their masking to assess foraging success. When they return, we can also look at their energy stores, their cortisol levels, their mercury loads to assess their health. And we can see how this affects their fecundity and survival. So with the elephant seals out at Año Nuevo, we have a long-term demographic data set, so we can start to understand how some of these stressors may impact populations. As I mentioned, the study is just ramping up. The seals are out at sea right now collecting data, but one of the kind of first things that we noticed this year is that the animals that we gave that ACTH to, so we elevated their cortisol levels, they stayed on the beach longer before they weaned their pups. So this figure is probably a little overwhelming, but we have lactation duration on the X, weaning mass on the Y, and then the size of the circle is female age. So the big circles are older females, the smaller circles are younger, and the animals that are in blue and pink are the animals that were given that ACTH, and green and purple were not. So at this point, the acoustic tag doesn't really matter because they hadn't gone to sea. And already, just by elevating their cortisol, we can see that they extended their lactation duration by 3 days. But even though they stayed on the beach longer nursing their pups, we assumed they did not wean larger pups. So basically we found that this elevated cortisol is affecting life history patterns, but we don't really know the mechanisms yet. So we're starting to look a little bit more at that. This is, again, just work from this February. So to kind of wrap up what I've been talking about today, we have found evidence that human interactions can be energetically costly. What we observed in sea otters is that there was relatively minor costs, but we need to do more work to see some of those hidden costs. We have evidence that acoustic disturbance extends dive duration. This has the, potentially will make them less efficient foragers, but it also could potentially alter how they manage their oxygen and their nitrogen, putting some species at risk to decompression sickness. And then just some preliminary results from our elephant seal work is it looks like some of this physiological stress, even just after a few days of having elevated cortisol, can modify their life history patterns. And we're using all of this information, kind of going back to that population consequences of disturbance model, to try to look at how these changes in stress level and heart rate and foraging behavior are impacting the health and the vital rates of these animals, so we can better predict how these disturbances are going to impact populations. And then just as kind of a fun aside, we just this year submitted a proposal to also see how personality or animal personality influences some of these physiological and behavioral responses to stressors. So we've kind of all noticed when we're out there working, animals tend to have different personalities. And so we're starting to assess that more systematically and categorize the animals as either bold, kind of assertive, cautious, and see how some of this might influence how they respond to disturbance. And with that, I would like to acknowledge kind of everyone that helped accomplish all of this research. So most of the collaborators were listed on the specific projects, but these are other people that have provided data or mentorship to my students. I also, of course, want to acknowledge my students who have helped do a lot of this work. The students listed here are the ones that specifically helped with the projects I'm talking about, but we also work a lot with other labs. For example, right now, I'm in Rachel Holzer's lab office because I'm in the field and don't have access to mine. And then we've had numerous funding sources: Office of Naval Research, NSF, Department of Defense funding, and then couldn't do this work without the support of Año Nuevo Reserve, and some of my students have gotten funding from Myers Trust. And all of the otter work was in collaboration with Sea Otter Savvy. And with that, I'm happy to take any questions.
Thanks so much. I don't know if I'm hearing an echo. Okay, thanks so much, Brigitte, for sharing your research with us. I want to echo a comment that came up in the chat with someone sharing that he's just so impressed with the multidisciplinary skill sets that these teams of researchers have to engage in this work. And I'm really blown away by what we learned, and also by that really cool animation showing us that dive. I wanted to start opening up Q&A with the time that we have by actually injecting a question of my own, which is, I'd be really interested to hear a little bit about I appreciate you sharing how you started off on this pathway as a researcher at the start of your career, choosing an area that allows you to bridge multiple different areas, subfields that you found really interesting. And so I just wondered, how has your research changed over time, given the increase of the impacts in climate change that we've seen? Like, how has it impacted your styles of research or the way in which you have to go about this work?
Well, that's a great question. I will admit I started off probably more as a basic scientist, and I still do see the value in that. A lot of times I just wanted to know how things work, like how is an animal able to do that? But as we're kind of seeing the level of disturbance increase and the effects of climate change, it's just starting, okay, yes, we have to understand how they work, but now we can start to use the, like we can add these experimental disturbances and see, well, how are they able to respond? So I would say it's made I've started to make my work a little bit more applied, but I never really want to fully leave that basic science, because I think you just have to know how things work in order to know kind of how it's going to change. But I definitely, I started off probably more as a basic, just wanting to know how things work, and I'm starting to ask more applied questions as time has gone on.
Thanks. Here's a question that says, your most recent research into animal personality sounds great. I'm curious about your approach. Is it at all informed by how humans have historically studied human personalities?
It's a little bit different. I'm sure it has some of the same things, but basically personality is defined as this, kind of a consistent response to a new stimuli. So in marine mammals, how that's often assessed is through things like novel stimuli assessments. So you show an animal a novel stimuli and you kind of judge how they react. So you have experienced observers, like right now we're in the process of kind of creating our behavioral spectrogram. Does the animal approach? Does it run away? Does it acknowledge it? And we're kind of coming up with the ithogram, like what are the possible responses? And we're doing this, so you need to do it multiple times on the same individual to kind of find out what is this consistent response. And we do find that some animals you show them something new, they might go and attack it. Other animals, you show them something new and they kind of run away. And so we're at this point, we're still doing more test trials to figure out kind of what is going to be our behavioral spectrum. But that's the way that we're going to do it is this novel object. So showing them a new object and how do they respond to that, and then kind of is the response. So the step one is, do they show a consistent response? Are we going to be able to use this as a tool? And it does, for many of the kind of pilot studies, it does look like it is a consistent response.
Great, thanks. There's a question here that's asking whether all experimental acoustic disturbances were killer whale vocalizations, right? And if so, how do those results kind of compare to more anthropogenically caused noisier oceans? How do we correlate those responses in the animals?
Yes. So in this case, we're using killer whale mainly because we wanted to be consistent, but the previous study exposed the animals to different sounds such as sonar, and they found that they have the same, they didn't measure physiology, but they have the same dive reversal. So we chose to use kind of a natural sound at this point, even though, just kind of how do they respond to that? In future years, we are hoping to change that to sonar to see if it's different, but just based on their behavior, we do know that they respond the same way to sonar as to this killer whale. But we don't know exactly, maybe their physiology is a little different, but we suspect it's very similar.
Okay, there's two questions here that go back to the sea otter, the kind of first example that you walked us through in your presentation. And one is, are there some mathematical models for the distance disturbance that you were examining? And then another question is, can you please talk a little bit about the hidden Markov models that you use to classify sea otter behavior?
Yeah, so I should get my graduate student on here. I will admit she worked with Tim Tinker to do most of that modeling. But hidden Markov models, kind of the theory behind them is trying to see sort of hidden states. So we're seeing some observations, and I should pull up those slides. But we see some observations and we're trying to kind of predict, well, what was the state before that, or what caused that? And someone kind of gave me a good example, like If you're trying to predict the weather, where someone is, they have three common behaviors that they do, and so if someone's going for a walk, that usually means that there's good weather. If someone's staying indoors watching movies, that's, typically means that there might be a rainy day. So it's not you're observing some behavior and trying to predict sort of this hidden state that you can't see. So that's that type of modeling, that hidden Markov model. And so, yeah, like I would say, I should be able to answer that better, but kind of use that modeling. So based on all of these observations and what the environmental conditions are, they were able to say, okay, based on this, stimuli was here, how did the otter respond? At what distance did that otter respond? And they just used hidden Markov models as their tool. And I do have slides in a different talk with more of that detail. And so I admit that that's a horrible explanation, but my student did all of the math for that project.
Another question here is whether, sorry, my thing is jumping around. Can you talk a little bit about the process of tagging and some of the stresses that may cause on the animals in doing this research?
Yeah, that's a great question. So every animal is kind of tagged in a different way. These sea otters, we did not instrument. Those were all behavior observations. So we did not influence their behavior there. With the elephant seals, one of my graduate students who's going to be defending next month, we really wanted to see what kind of effects that we have. So we measured heart rate and hormone levels and some metabolites throughout our entire kind of anesthesia procedure, recovery, and then when they returned on the beach. And of course, there is a little bit of stress associated with when we have them on a table and we're doing our procedures. But basically, when the animals were recovering in the cage overnight, those levels dropped pretty significantly. And by the time we recovered them at the end of the translocation, they were back to baseline levels. So basically, what they were on the beach right when we drugged them, they were the same. And so we do know that there is the potential that these instruments have effects, but at least with the elephant seals, it seems to be pretty minor. So we do our best anytime we do these types of studies to try to do some sort of controls like that. Look at survival of instrumented animals versus non-instrumented animals. But specifically with that elephant seal project, we did look at that, and we were really impressed. Like, you would think maybe being in a truck and driven down to Monterey might be stressful for these animals, but based on our heart rate records, it looks like they just fell asleep, or most of them just fell asleep, and you would see the typical apnea, like decrease in heart rate with apnea, with breath holds. So they're a pretty resilient study species. That's one of the reasons they're also great to work with for this.
Really interesting. Well, I wanted to say, unfortunately, that's all the time we have for questions, though we are saving all the questions and they will be shared with Dr. Mcdonald so that she can respond to folks who submitted things that we weren't able to address today. And with that, I just want to say again, thank you so much, Dr. Mcdonald, for sharing your really fascinating work with us. This has been a great talk, and we look forward to seeing more of the results coming out from your lab and the great work that you and your students are doing. Thank you, everyone, for attending today and engaging with us. And thanks again to the team behind the scenes that helps to make this happen. We wish you a great rest of your day.
Thank you very much.